All right, so my name is Jeff, and my partners Arden and Amali and I worked on a presentation about pedigree analysis, application, and genetics testing.
So our found paper was sex-linked barring in chickens is controlled by the CDKN2AB tumor suppressor locus, which was published in 2010. The paper shows that sex-linked barring is controlled by this locus, which encodes the INK4B and ARF transcripts. They identified two non-coding mutations in CDKN2A that showed near-complete association with the phenotype. They also discovered two missense mutations identified at highly conserved sites. Further work would be required to determine if one of these or a combined effect of two or more CDKN2A mutations is causing sex-linked barring. Their novel finding provided the first evidence that the tumor suppressor locus CDKN2AB can affect pigmentation phenotypes and shed new light on the functional significance of this gene. There are actually two types of feather barring inheritance, as it can be sex-linked or autosomal. Sex-linked barring produces bars with a complete absence of pigment, while autosomal barring produces bars that alternate between darker and lighter pigmentation, presenting as a more irregular pattern. And these are all hens, which are female chickens, exhibiting a feather barring pattern. Sex-linked barring, a common plumage color in many breeds, is characterized by black and white barred feathers. Previous studies had indicated that the white bands are caused by an absence of melanocytes in the feather follicle during the growth of this region. In wild-type feathers, melanocyte progenitor cells migrate up from the feather base and start expressing ARF in the barb region, leading to differentiation of melanocytes and pigment production without exhausting the pool of undifferentiated melanocytes. In sex-linked barred feathers, upregulated ARF expression may lead to premature differentiation of pigment cells and a lack of undifferentiated melanocytes that can replenish the ones producing pigments. As the feather keeps on growing, no more melanocytes are available to produce the pigmentation, and it creates the white bar. Sex-linked barring shows a monogenic inheritance, whereas autosomal barring is assumed to be caused by the combined effect of alleles at the dark brown and penciling PG locus. In birds, females are the heterogametic sex, ZW, and males the homogametic sex, ZZ. Sex-linked inheritance of sex-linked barring is BB, and the wild type is BN. The paper uses the official chicken genetics nomenclature, where the first letter B refers to the locus and the second letter B or N refers to the allele. The B locus shows a classical gene dosage effect. BB homozygotes display wider white bands than either BB-BN heterozygotes or BB-W hemizygotes. Other characteristic phenotypic effects of the B allele include the dilution of the dermal pigment in the shanks and beak and a white spot on the head, which is larger in males than in females. CDKs are cyclin-dependent kinases. They're expressed at a constant level throughout the cell cycle. CDKs act to transfer phosphate group. They are responsible for the shift from G2 to mitosis because of their phosphorylation of proteins. The next aspect of this concept is the CDKN2AB locus. The study shows that the sex-linked barring is caused by the CDKN2AB locus, also known as the INK4B ARF locus, which you can see in Figure 2. It encodes both the ARF protein, which binds the p53 stabilizing protein MDM2, and the INK4 protein, which inhibits CDKs, causing a cell cycle arrest in the G1 phase. In humans, CDKN2A encodes two transcripts, INK4A and ARF, while CDKN2B encodes the INK4B transcript. The CDKN2AB locus has been shown to be involved in tumor suppression and cell proliferation as ARF and INK proteins regulate the cell cycle.
All right, so the second paper that we all selected was a paper about population structure and inbreeding from pedigree analysis of purebred dogs?
So dogs and humans. Well, a big question that you may be asking is, how are dogs relatable to humans? And one of the reasons that you can compare these dogs to humans is the types of diseases that these dogs are getting. The first of those is cancer, which is obviously prevalent in both humans as well as dogs. All different types of autoinflammatory diseases as well have been found in dogs and in humans. And retinitis pigmentosa is a disease that is seen both in dogs and humans, which can make the dogs almost a model organism compared to the humans. And some of the researchers for this experiment were examining if specific diseases related to certain breeds. And they found that main diseases are only prevalent in one or a few breeds, which led them to be concerned about the different types of breeding patterns of dogs. So pedigree analysis. For this experiment, they used 10 different types of breeds, the Akita Inu, the Boxer, the English Bulldog, the Chow Chow, the Rough Collie, the Golden Retriever, the Greyhound, the German Shepherd, the Labrador Retriever, the English Springer Spaniel as well. And to analyze the different species, they looked at the different types of sizes of pedigrees and the complexity within those pedigrees, the distribution of the offspring, the inbreeding and diversity, as well as the population structure.
All right, so as far as the results and discussion go of the paper, they focused on the four categories that Arden just discussed. The first one being pedigree size and complexity. Generation numbers were seen to move from 5.9 to 9.0, with the 9.0 representing the German Shepherd population. The lowest rate of generation imbalances was seen in Golden Retrievers, which was of interest to the researcher, and we'll discuss more of why of that later. The next category that was discussed was offspring distribution. Akita Inu was seen to have the most even distribution of reproductive success, meaning that it definitely follows that random principle where the breeding isn't set particularly. The next part that was discussed was population structure, and the researchers found that a systematic structure was seen in the Springer Spaniel English breed, and they found this extremely interesting because there was some sort of subpopulation breeding going on that they found in another chart that we will discuss later. Let me go to the next slide. So as we can see here, there's two things that researchers really enjoyed to look at. We see that the number of dogs, the Labrador Retrievers, had the greatest amount of species, obviously, but yet had the lowest percentage of males for some reason. They found that of interest. They also were questioning the number of founders here for the Greyhound species, which also had the lowest amount of population, yet had one of the mid-row to higher percentage of females.
And so for this next table, it's showing the comparisons between the sires, offspring, and the dam offspring. And you can see the median numbers of offspring per sire dog of each species. And you can see that some of these sires are being used to generate thousands of offspring compared to the population of dams that are only being used to generate within 50 to 100 of offspring usually. Now the median numbers are around roughly the same, but the maximums is where it gets interesting for the sires, which could be related to different types of genetics and the lack of diversity within some of these species.
So as we can see here, the population structure, the main thing researchers were looking at was this strong line relationship here and also in the PCA1 relationship, meaning that there was some sort of subbreeding going on. We're running out of time here, so if we go to the next slide. This is the inbreeding and diversity chart. And the one thing the readers like to look at here was the Greyhound being so high in generation 5 and also 4. This includes all 10 species, but again, we're running out of time, so can we go to the next slide? And
comparing and contrasting the two different types of papers, we see similarities. Both of the papers involve pedigree analysis of the dogs and the chickens, and they both discuss the inheritance of genes with both papers. The differences would be that the classic paper talks about sex length and the select paper does not, as well as the multiple species covered in the found paper and only chickens discussed in the classic. And the genotypes were discussed more in the classic paper as compared to the found paper.
And that's it.
Questions?
Right now.
References?
Questions and some references.